All right, so for this first section on nephron physiology, we're going to talk about how exactly that filtrate is formed. So primarily we're going to be focusing on that renal corpuscle with the glomerulus and the Bowman's capsule and how that filtrate is formed. Okay, so first of all, let's go over the filtration membrane and what exactly that is. So the filtration membrane in the kidney consists of the different structures that the filtrate, there we go, must actually pass through in order to become the actual filtrate, right? To get to that capsular space. Okay, and so those structures are, if we start in the capillary, right, it'll first have to pass through the simple squamous, right, of that capillary. So that would be here. Okay, so that filtrate is going to have to pass through that simple squamous cell. And then the basement membrane of the capillary. So remember from A&P1, your epithelial cells, right, they all on the basal side, the bottom side, have that layer of glycoproteins called the basement membrane. So that filtrate is going to pass through the cell itself and then through that basement membrane, and then it's going to reach those podocytes that are the visceral layer of the glomerulus. Okay, and so those podocytes, the filtrate is not actually going to pass through the cell itself. It's going to pass through these little gaps in between the feet of the cells, and those are called the filtration slits. Okay, so that filtrate has to pass through these filtration slits between those podocytes. Okay, and so as you can see, that would kind of limit the size of the things that can actually leave those capillaries, right? So the things that leave have to actually be small enough to fit through those filtration slits. So when we're talking about the composition of the filtrate, the composition, it is made from the blood, so it's going to be very similar to plasma. It's just not going to have many proteins, right? It might have some very small polypeptides, but there aren't going to be any large proteins, or there shouldn't be any large proteins in urine, okay? And if you do find that somebody has protein in their urine, that could indicate that there's a problem with this filtration membrane, right? This delicate membrane has maybe been damaged, allowing large things to escape. Okay, so it's going to contain a lot of stuff like glucose, can easily move out the amino acids, a lot of ions, and then of course those nitrogenous wastes like urea and ammonia are going to be removed. Okay, all right. So let's talk about how filtrate is formed. So filtrate is formed, first of all, through the process of filtration. And we're going to kind of define that word again in a minute. I'll add it here, though, because this is a good place to talk about it. So filtration is just when molecules are moving from high to low pressure. All right, so it's different from diffusion, which is high to low concentration. Here in the kidney, we're using pressure to form that filtrate, to force those things out of this capillary and through those filtration slits. Okay, so from high to low pressure, and we've talked about filtration before very briefly. Okay, so let's talk about the pressures that are involved. So the first pressure that's involved is the blood hydrostatic pressure. This is essentially just the blood pressure within the capillary itself. Okay, and so it's going to be pushing out of the glomerulus. And typically this pressure within the glomerulus, these special capillaries, is going to be much higher than in a systemic capillary bed, which we've talked about before. So that's usually around 25 millimeters of mercury in a systemic capillary bed. Remember, it changes across, between the arterial and the venous side, but kind of on average around 25. All right, so this would be systemic compared to our glomerulus, right? And remember we said how important that pressure is to forming urine, so it does need a higher pressure. And so that pressure can be changed by altering the diameter of that afferent arteriole. Okay, and so let's say you want to increase the pressure in that capillary bed. All right, what you can do in that glomerulus, you can make this afferent arteriole, the diameter, wider than the efferent arteriole, and so then you're allowing more blood to enter that glomerulus than is able to leave through the efferent arteriole. Okay, so then you have a higher kind of volume of blood entering, and it's not as easy for it to leave, so that'll increase the pressure. So you can kind of adjust the diameter of this arteriole, right, because it has a lot of smooth muscle, to change that pressure. And so that would be the blood hydrostatic pressure. And then you have the capsular hydrostatic pressure. So this would be the pressure pushing in on the capillary. And that is simply the pressure from the fluid, right, that filtrate that's building up in that capsular space. Okay, so the more filtrate that builds up, that fluid is going to push in on the glomerulus, right, and kind of counteract this blood hydrostatic pressure. Okay, and then lastly we have osmotic pressure. So osmotic pressure, we've talked about many times before, is from typically albumin, right? This is going to be fluid being pulled in towards that albumin. Okay, and then we can talk about glomerular filtration rate. And so this is just the rate that filtrate is being formed at. And so typically in the kidneys, it's about 180 liters a day of filtrate being formed. And so obviously you should be thinking, well, we certainly don't make that much urine every day, right? Otherwise, we would be, having to urinate constantly. So a lot of this is going to be reabsorbed, this water, okay? But typically about 180 liters of filtrate is formed each. And so this rate of forming the filtrate, this glomerular filtration rate, should be maintained constant throughout the day, right? And so there are mechanisms in place to help maintain that filtration rate at a constant level. And so let's talk about those mechanisms. So the first kind of category is going to be autoregulation. So this is going to be the kidney kind of regulating itself. It has its own mechanisms in place to regulate that filtration rate. Okay, so autoregulation, the first one is the myogenic mechanism. All right, so myo should tell you that muscle is going to be involved somehow. And so this is going to occur in the afferent arteriole. And so remember we said that afferent arteriole is often drawn really thick because it has a lot of smooth muscle. So one thing that smooth muscle can do is it can detect stretch, right? So stretch from an increase in pressure. And when it detects that stretch, which would indicate an increase in blood pressure, that arteriole is going to respond by contracting. Okay, and so the reason it does this is to kind of, in a sense, it protects the glomerulus from that increasing pressure. Okay, because remember that glomerulus has that very kind of delicate, thin filtration membrane that you don't want to get damaged. And so if that blood pressure goes too high in that glomerulus, you can damage those little membranes. So this mechanism will help to kind of maintain that pressure so it doesn't get too high and damage the membranes. And then obviously the pressure has an influence on the filtration rate as well. Okay. And then our second form of autoregulation is going to be called tubuloglomerular feedback. Okay, and so this involves that juxtaglomerular apparatus. So the distal convoluted tubule part of the juxtaglomerular apparatus contains specialized cells, right, called macula densa cells. And so these cells are going to detect how much salt is in the filtrate. Okay, and so essentially the levels of salt in the filtrate that's moving through that distal convoluted tubule, remember that's right before it goes into the collecting duct, right? So this is kind of the last stop before it leaves the nephron. So the salt in there will tell the kidney something about how the filtration rate is. Okay, so if the salt levels are really low, that filtration rate is too slow. And so it's too slow because it's allowing too much time for reabsorption to occur. All right, so it's going so slow that those mechanisms that reabsorb all the ions, the salts, are able to pull out way more salts than they should be. Okay, so low salts means that it's too slow. And so the response to that will be to reduce constriction of that arteriole and allow more movement, right, through that area, right? So that'll increase that filtration rate and get things moving a lot faster. Okay. All right, so that's autoregulation. So then we have some hormones that will regulate the filtration rate. So we've talked about renin because of its importance in regulating blood pressure. So it can increase blood pressure, thus influencing the glomerular filtration rate. And the heart can actually influence the glomerular filtration rate through atrial natriuretic peptide, right? So it's made in the atria, and it regulates kind of the amount of salt in the urine, okay? So it will actually, releasing this hormone will cause the urine to be more salty, okay? And so it's released because there's too much salt in the blood. And so it tells the kidney you need to get some of that salt out of the blood and put it into the urine. So it makes the urine more salty. All right, and so therefore it's going to increase that filtration rate. Okay, and then we also have the autonomic nervous system playing a role through the sympathetic branch. So that sympathetic branch, when it's activated, it's going to constrict that afferent arteriole, and it's going to decrease the glomerular filtration rate, because we know the sympathetic nervous system kind of shuts down all those rest and digest functions, which include the urinary system, right? So that's going to decrease the glomerular filtration rate. All right. So now let's just go over a few kind of review terms before we get into the different functions of the different tubules that the filtrate is going to pass through once it leaves that renal corpuscle. Okay, so our first term is filtration. We just talked about this. So filtration is just the idea that solutes are going to move from high to low pressure, right, instead of high to low concentration as it would with diffusion. And then we talked about reabsorption. So reabsorption is when something is taken from that urine or that filtrate and it's put into the blood, right? These would be things you want to keep. So they end up in that filtrate, like glucose and amino acids and some ions, and you want to put it back into the blood because we want to keep it. Secretion would be things we want to get rid of. So we're going to take those things from the blood, like waste products and sometimes potassium and hydrogen ions, and we're going to put those into the urine or the filtrate. Okay, in the next section we're going to be talking a lot about kind of exchanging different ions and glucose and amino acids, so there's going to be some transporters involved. So let's just review some terminology for that. So a co-transporter is going to be a transporter, so this would be a membrane protein, and it's going to move two molecules, and it's going to move them in the same direction. Okay, so co-transporter moving two molecules in the same direction versus an antiporter, which moves two molecules in an opposite directions. All right, so you'll bring one molecule in and the other one out. Okay, so then there's going to be two types of reabsorption. Primarily, we're going to be focused here on the transcellular reabsorption. So this just means that molecules like glucose and amino acids are going to move through the actual cells of the tubules and into the blood, versus pericellular reabsorption. They would move kind of between the junctions of the cells into the blood. Okay, so typically we're going to be looking at transcellular reabsorption.